Voice check.
Voice check.
Voice check.
Hello, everyone. Welcome to 11th Journal Club for Antibuddies. And today we have a special guest with us, Dr. Scott Walsh from McMaster University in Canada. Hi, Dr. Walsh.
Hello, thanks for having me.
And as you guys might have seen from the post yesterday, Dr. Walsh is a research associate in Dr. Wan's lab, and he has some very cool work with oncolytic viruses and adoptive cell transfer. Dr. Walsh, just before we start, can you tell us something about what got you into this area of work?
So it's actually an interesting progression. Um, I myself am diabetic, which is an autoimmune disease. And so growing up, I've always been interested in research related to the immune system and how the immune system can basically turn on itself and attack its own tissues. And so I ended up in this area because viruses are a very prominent way to activate the immune system and have been connected to autoimmune disease. At the same time, immune system attack of tumors is inherently an attack on one's own tissues too. So it really helps us to understand how this barrier can be crossed when it comes to the immune system and autoimmune disease.
That's pretty nice. A lot of people I've noticed that maybe it's just a bias for me, but I've noticed a lot of people who are diabetic end up in research, or maybe I just know too many people who are diabetic and they have ended up in research either with autoimmunity or actual diabetes, type 2 or type 1. Pretty nice. Hey, Dara, can you hear us?
Yes, I can. Hi, everyone.
Did I pronounce your name
Hi.
right?
Yes,
Okay, so
that's
Dara
correct.
is our newest member, and we haven't got to speak at all. So, Dara, can you tell us a little bit about yourself before we start? Because
Okay, I'm Dara. I'm a master's student in Paris at University of Paris-Saclay. Yes, I'm doing biology. Nice
nice.
to meet everyone.
Okay. Jait, before we start the discussion, just for everybody that we'll try to ask questions. If there are things you don't know or maybe you think would be useful for the audience, audience is mostly graduate students and undergraduates. So if there are some basic concepts that could be very useful for them to understand, we will try to talk about them. And with that, Dr. Walsh, the stage is yours. You're free to share your screen.
Alrighty. All right. So as you mentioned, my name is Dr. Walsh, and I'm a research associate in the WAN lab, and we are in the McMaster University within the McMaster Immunology Research Center. And this is some of the work that we've been working on today that I'll be presenting. So we kind of specialize in combining the use of adoptive cell transfer, which I'll describe later on, and oncolytic viruses, which again I'll describe later on. Work so that they can work together to create a better therapy for tumors. So what I'll be presenting today is the work from this paper here, which was published in the Journal of Clinical Investigations just over a year ago now. But before I get into all the nitty gritty, I understand that the purpose of this podcast, or one of the goals of this podcast, is to make the information that we're presenting accessible to a wider audience. So I'm going to spend more time on the introduction than usual and present more of a summary of the results of the study. So like I said, in the WAN lab, we kind of exist in this little pocket here where we study the overlap between cancer, virology, which is the study of viruses, and immunology, which again is the study of the immune system, and how they all individually interact. But really, we really like this specific intersect here. So to better understand how cancer and the immune system interact, I'd like to first go over how viruses and the immune system interact so that we can get a better comparison, because viruses and cancer are very, very different kinds of pathogens. But first, the reason why we study cancer is far too easy to justify these days. Cancer is a very prominent disease, making up a third of the causes of death in Canada, and that is just too high of a number to tolerate. So we do have existing therapies for cancer, which include chemotherapy and radiation. Those are the classical methods of treating cancer. But the list of side effects of these treatments is astounding. It's very long. Most people are aware of the hair loss and the fatigue and the nausea associated with them, but the list is actually very, very long. And it's, I think, just tells us that we need to come up with better treatments for treating cancer. And taking into account that what we've seen in the world, all the effort that's been put forward to help control the spread of COVID, it's still at this point lists below cancer as a cause of death within, this is in the States, but the same is true for Canada as well, and worldwide, I believe. But considering what the world has done to help spread the effort that's gone in to help stop the spread of COVID, I think we also need to, once COVID has been tackled effectively, we need to put some more effort towards cancer as well. And I think we can. I think that this has demonstrated what we can do as a global community. So this is what I was alluding to when I said we need better cancer therapies. It's been a while in the development, but within the last little while, cancer immunotherapy, the last 2 years, has emerged as a prominent and very useful treatment for cancer. So this is simplified as just basically training the immune system to target and destroy tumors through one way or another. And it's recently reached recognition back in 2018. The Nobel Prize for Physiology and Medicine was awarded to James Allison and Tatsuko Honjo, who are very prominent leaders in the field of cancer immunotherapy. And through their efforts, we've seen some very effective treatments reach the market, but there's still a lot of room for improvement. And that all comes back to this little statement here that the tumor, or a tumor, is a very unique pathogen. It's not the same as what we've seen in bacteria infections or virus infections. It's a disease that is very unique to its own patterns. So to give a little background on how virus immunity works, so we can better compare virus immunity to cancer immunity to show what the differences are and how they can benefit each other. So when a person becomes infected with a virus, the virus comes in, enters the cell, and starts replicating. So it basically takes over the cell and uses all of the raw materials and machinery in that cell to make more of itself. And by doing that, it replicates and ends up lysing the cell and spilling all of those new virus particles, although not all viruses lyse cells. They do release virus particles into the environment, and this then results in activation of the immune system. So this is done through several different pathways, but the moral here is that you have immune cells that detect the virus infection and become very inflammatory, which means that they release a lot of small molecules that cause the immune system to react. Just similar as an alarm bell going off and a house alarm going off would activate and recruit the police. The immune system is activated and recruited to the site of infection. And so this is characterized by inflammation, which most people associate with swelling, but at the same time, there's many other things that inflammation does, which again includes the orchestration of the immune response. And this also makes accessible to the immune system something we call antigens, which are just little pieces of that virus that the immune system can then react to. So effectively, it's taking a piece of that virus and putting out an APB to the immune system, with the immune system being the police force here, so that we can find in the draining lymph nodes, which are special structures designed for housing immune cells while they wait for something to do. So these DCs here take those pieces of the virus back to those lymphoid structures and basically try to find an immune cell that can recognize those specific pieces of the virus. And once that's done, you develop a robust response. And so this is usually done in two parts: a B cell response, which is responsible for making antibodies, which most people are very aware of. But on the other side, you have the T cell response. So these T cells are responsible for identifying virus-infected cells and killing those infected cells, thereby not allowing the virus from producing more of itself in that cell. So basically, you've cut off the ability of that cell to produce any kind of new virus and limited the spread of the virus. So although antibodies can also be useful in tumor immunology, we're going to focus on T cells today because that is the focus of our study today. And so we'll focus on them moving forward. But in the context of virus infection, like I said, these T cell responses are developed, recruited to the site of infection, and they kill off the virus-infected cells. At the same time, and that usually takes about 2 weeks for the virus infection to be resolved. And so that's actually part of why the quarantine period for COVID or suspected COVID infections is about 2 weeks, because that is about how long it takes for an immune system to find and destroy the virus infection. But after the virus has been cleared, we develop something called a memory response. So there's those same cells that could recognize the virus will persist in those lymph node structures, waiting for a secondary exposure to that same virus infection, so that when it does happen, they can respond much quicker and eliminate the virus threat. And so this is actually the point of getting vaccinated, is to develop a existing memory response that will wait in the lymph node for your exposure to that virus, and therefore prevent it from infecting you effectively and allowing you to spread to other people. So how does this differ from tumor immunology. So the first difference is that, like I said before, tumors are derived from your own cells. So there's nothing very inflammatory, or there's not very much inflammatory reaction involved with that. And there's not a whole lot of targets available for the immune system to identify that are unique to the tumor and won't cross-react, won't allow those immune responses that developed to attack normal tissues. But because tumors are very mutagenic, they like to change very quickly, so that their DNA will change in order to allow them to do things, typically proliferate quicker. They will accumulate a certain load of mutations. So this is small changes in the genetic code that result in a change in one or more of their proteins. So it just being a normal protein here, small change happening, so you end up with a little yellow dot here, and the immune system can take advantage of that small change to then identify and target the tumor, because this mutation is very specific to the tumor and provides a very specific target for the immune system to use for identifying and destroying tumor cells. So just like with virus infection, it is the responsibility of DCs to find the tumor cells, and typically you do have some death of tumor cells occurring during growth of a tumor. This is due to several different factors, but typically they outgrow their nutrient pool and some of the cells die from starvation or other mechanisms. But when this happens, it happens in the absence of inflammation. So these DCs still find those pieces of the tumor and take them back to the lymph node, but they don't have that same inflammatory signal to tell the immune system that, we need to pay attention to this and we need to come back to the tumor and destroy it effectively. So you still do have a T cell and B cell response develop, but the magnitude of that response is much lower and typically isn't as efficacious as it is for virus infections. So typically at the tumor, you can develop a natural tumor response, but they are typically ineffective, and that's actually very observable in the fact that we develop tumors. If they were effective, you would never actually see that tumor develop beyond the initial growth. So, and the barriers to this are typically, like I said, low inflammatory signals, so that results in a too small T cell response, and those T cell responses that do develop don't get brought into the tumor effectively. So these T cells can't kill tumor cells if they never actually see those tumor cells. So they need to be brought into the tumor to see them. And there's also several other mechanisms that tumors use to suppress T cell responses once they do reach the tumor. And part of that is preventing those T cells from getting into the core of the tumor. So if you do get some T cells that manage to make it to the tumor, they typically get stuck on the periphery of the tumor and don't get into the core to allow those T cell responses to completely obliterate the tumor. So knowing what we know about viral immunity, what we like to do in the Wand lab is to try to convert tumor immunity into a more viral immunity-like situation, because we know that viral immunity works very well, and a lot of the deficits of tumor immunity can be addressed by those specific, especially the inflammatory aspects of viral immunity. So to do this, we like to work with something called oncolytic viruses. So these are a specific group of viruses that, for one reason or another, prefer to replicate in tumor cells. There are several reasons for that, but for the virus that we'll be discussing today, which is vesicular stomatitis virus, or VSV, it is very sensitive to the internal methods that normal cells use for shutting down virus replication. And in tumor cells, those methods or those mechanisms are typically dispensed with in favor of allowing the tumor cell to proliferate really rapidly and really well. So because of that, these viruses can replicate really well in tumor cells, but not so well in normal cells. So by administering an oncolytic virus, such as VSV, we can allow these tumor cells to be lysed by the tumor growth, which is basically just breaking that tumor cell open, and exposes more antigens, more neoantigens, like I said, the mutation-derived special proteins associated with tumors, and also inflammatory signals too. So this helps to make that tumor cell death more like a virus-induced cell death. And so this then allows us to recruit DCs to the tumor, pick up those neoantigen particles, take them back to the lymph node structures, and allows for development of a much more robust T-cell response. But at the same time, and it's, I think, underappreciated in scientific literature, oncolytic viruses can still, or some at least, can still enter normal cells. They may not be able to replicate well in those cells, so they typically don't result in the lysis of those cells and production of progeny virions, or I guess baby virus particles. But they can actually enter those normal cells and can express some of their proteins, some of their genes. So we like to take advantage of that by using a two-pronged assault, where we allow this normal virus infection or normal cell infection to provide a vaccine effect, which we'll get into in a second or so. So this is demonstrated well with this picture here. This is a picture of a lymph node structure. That's actually the spleen. So there's little follicles within the lymph node where the B and T cells preside. And around that is an area of dendritic cells and macrophages, which are those special cells that like to bring the virus pieces, the tumor cell pieces, to the T cells and B cells to activate them. So we've engineered our oncolytic virus here to express GFP. So it makes our cells that are infected glow a green color. And you can see that we have a lot of cells, normal cells so they're not tumor cells, but they're normal cells that are infected and expressing this GFP protein, but in the end, they will not be killed off by the virus infection. So we can make use of this by using our oncolytic virus as a vaccine as well as an oncolytic component too. So in this way, in addition to infecting tumor cells and making tumor bits available to the DCs and inflammation available to the DCs, we can also directly infect those same DCs in the lymph node to give us a double punch for the stimulus of these tumor cell T-cell responses and recruitment back to the tumor. But like I said, we still have that issue where the T-cell responses are not very robust as generated by the tumor growth. So there are some T cells here that were generated during tumor growth that the virus infection can stimulate, but we like to maximize on this effect by transferring T cells that we've generated outside the patient to maximize the presence, the number of tumor-specific T cells in the lymph node. So we do this through something called adoptive cell transfer. So first question is, what is adoptive cell transfer?
I had a question, Scott, sorry, from
Yep.
the previous. So this oncolytic virus, can they also target the tumor cells which are at the very center, or even they mostly target the peripheral cells?
So it depends a bit on which virus you're using. So the funny thing is that oncolytic viruses, they can be there's a distribution of them across most different kinds of viruses. So it's a whole study of itself to find and identify why oncolytic viruses are oncolytic. But so it depends a bit on which virus you're using, but typically, and the route of injection too. So you can directly inject the virus into the tumor. So that allows you to have more virus infection, but that would then truncate how much infection you have here. So with the virus that we use specifically, and because we use intravenous injection specifically, we tend to see infection of the tumor, which starts around the periphery of the capillaries, the veins that go through the tumor, the blood flow, blood vessels. And they get a certain amount into the tumor, but yeah, they don't typically get into the core of the tumor. Because they are their fitness is low, so that's what makes them not very good at infecting normal cells, but better at tumor cells. They do, after a couple rounds of infection, you see a suppression of the virus infection.
Okay.
So they get in, but not very far.
Mm-hmm. Thank you.
So to generate T cells specific for our targeted tumor antigen, so that'll be the same neoantigen that we encode in our virus backbone, we can isolate tumor-specific T cells from a patient. So you take them out, you put them into a dish and something we call in vitro culture. And by specifically stimulating them with that neoantigen that we're targeting, we can make them proliferate and grow up to a very large number and a very large functional capability, so that we then reinfuse them back into the patient. So we've taken out 100 cells and we're putting back in 1 billion cells. It's a big change and allows those T cells to have a very robust effect. But when it comes to combining this with our oncolytic virus, we know from previous studies that memory T cells are the better T cell type for boosting with our oncolytic virus. So to go back to our virus infection, our virus immunity slide, these cells, the effector cells here, are the ones that have the best ability to kill target cells. And so those are the ones that arrive at the tumor or arrive at the infected tissue type to kill off the virus infected cells, but they don't have very much ability to proliferate. So that's why they don't combine well with our oncolytic virus, because they don't end up surviving very long. But with using central memory T cells, which are those same cells that survive past the initial infection and are able to respond again to the secondary encounter with the virus, we're able to contain or to capitalize on their proliferative potential. They're able to survive very long and proliferate rapidly upon stimulation, and then they differentiate into effector T cells. So we're able to transfer in central memory T cells, and then through oncolytic virus vaccinations, subsequently convert them into a large number of very cellularly functional effector T cells. So with our working model, we use this paradigm where we can capitalize both on the transfer T cells through our oncolytic virus vaccination, and then provide a secondary boost of either transfer T cells or cells that have been existing that are generated by the tumor growth itself through the oncolysis component of the therapy. And then both of these cell types would be recruited to the tumor through the inflammation induced through virus infection of the tumor cells. So that allows us to maximize the tumor-specific T cell response and also the tumor effect. So for this study, we used the CMS5 model, which is a cell line mutagen-induced fibrosarcoma, which just means that it's a very dense, solid tumor structure. But the reason why we use them is because they are well characterized to express a neoantigen due to a mutation in the ERK2 gene, which I'll refer to as the ERK-M, so ERK mutant in this study here. And usefully, there's a strain of mice that have been engineered so that they have T cells specific for this ERK-M protein, this ERK-M neoantigen. So we can take cells from the duck-18 mice, grow them in culture to make them central memory, and then infuse them back in to CMS5 tumor-bearing mice to see how those T cells can then attack the tumor. I just want to note here that duck-18 mice express a marker. So this is just a little protein that we can stain for that will allow us to differentiate transferred cells, which will be the Thi-1.1, compared to endogenous cells, which are Thi-1.2 positive. That'll be useful later on, but the point being that we can then differentiate those cells, and endogenous cells are those cells that are generated through tumor growth in those mice before we even transfer any T cells in. So by using these two components, we can then combine them with the VSV that is expressing this ERK-M mutation here too. So we have oncolytic virus expressing our neoantigen to be used to boost those duck-18 T cells.
Hi, Scott. Can I ask something?
Yeah.
First, I have two questions. First, what's unique about VSV that makes it a good oncolytic virus?
So it's actually not the best oncolytic, but it's a very good vaccine. So
Okay.
that's why we kind of like it the best. So like I said, it relies on tumor cells and their inability to react to virus infection that makes them oncolytic. But not all tumor cells are completely absent in these specific mechanisms. So they can replicate in tumor cells better than in normal cells, but their replication in tumor cells is not as good as some other oncolytic viruses.
Okay.
But like I said, they are very good. They're very inflammatory. So those inflammatory signals I was talking about before, they induce a whole whackload of those, especially compared to some other oncolytic viruses. So we like to use it because it is very intrinsically inflammatory and therefore very good at inducing T-cell responses against tumors.
Does wild type VSV infect mice?
Wild
Is it?
type? Yes.
Yeah.
Yes.
So what
So it's
did they get disease? Or?
they can. It depends on the dose, but typically VSV is so poor. Even the wild type virus is so poor because it derives from farm animals.
Okay.
So some of the things that it's developed to deal with the infection of farm animals don't cross to mice, and so it can infect mouse cells, but it still doesn't infect it very well. So you can overload the system by giving them billions of virus particles.
Okay.
But we use much less than that here, so that we haven't seen much toxicity associated with the virus infection itself, unless you use immunodeficient mice, but that's coming up later on.
Yeah. So, and this ERK gene, is this the same MAP kinase pathway ERK?
Yeah,
Okay.
ERK2.
So
Yeah,
that's probably how it's over-proliferating these cancer cells.
Yeah, so yeah, that's the funny thing. It's been shown that this mutation is a tumor driver mutation, which just means that it drives part of the proliferation associated with the tumorigenicity. So we thought, and this is a little bit of foreshadowing, that it'd be good to target this gene because there'd be no way that these tumor cells could lose the gene because they would need it to maintain their tumor status. So we thought it'd be a great model to see the best anti-tumor effect of our therapy.
It's very smart.
But like I said, there's well, sometimes you make the best plans, but they never really turn out. But it does sometimes result in a very fun story, like this one. So when it comes to treating mice, we have to first implant them with the tumor. So the CMS5 cells are implanted here, and then 7 days later, once the tumor's grown up to a palpable size, which means it's something we can actually see and measure, we infuse T cells, and those T cells go in and effectively go to the lymph node, but don't do much else beyond that, unless you follow up 24 hours later with an injection with the oncolytic virus vaccine. And then from then on, we follow the tumor volume, so we can see what the tumor grows or gets smaller, and the immune response. So just wanted to note here that how we evaluate the tumor response is by stimulating cells from the blood of these mice with that specific peptide, the neoantigen, that ERK-M protein, and to see how many T cells produce interferon gamma, which is one component of inflammation that is known to be produced by T cells upon activation with their specific targets. So we can, by seeing how many produce interferon gamma, we can tell that all these cells are specific to our ERK gene, our target, and we can evaluate how many T cells we see. And again, we can also use Thy1.1 versus Thy1.2 staining to identify the transferred cells compared to the non-transferred cells, the endogenous cells. So when we use this combination therapy, we first saw that PBS treatment, which is our control to show that without any kind of treatment, the tumors grow very rapidly and result in the death of the mice quite quickly. When you use T-cell transfer alone of our central memory cells or virus infection alone, you see very little benefit, and there actually is no statistical significant difference with the survival of these mice. But when you use the central memory T cells in combination with the oncolytic virus vaccine, you see very robust regression of the tumor and followed by no relapse whatsoever. So these mice, we followed out for at least 60 days, and we saw no evidence that the tumor was going to come back at all, which is really an example of how synergistic this combination is by putting in T cells and combine that with the virus to activate them and recruit them to the tumor, we'll be able to have the maximal anti-tumor effect.
Can I ask something? Isn't
Yeah.
so when I was looking at this, I reread this whole figure about 3 times because I
Yeah.
could not believe that there is 100% survival. Like, did you guys
Yeah.
go out and party outside after getting these results?
Actually, this wasn't. This is the second time we've used this model, so this wasn't actually surprising. We've done it previously. Like I said before we got on air, I have another JCI paper that was published about 9 months before this one, wherein we had used a different model to show the exact same effect. So I think it just demonstrates that even among different mouse strains, different tumor models, we're able to have this same robust anti-tumor effect, and it's very consistent.
Yeah,
So
that's too
yes,
good. That's very good data.
it's almost too good.
I was just surprised that you got 100% survival till 60 days. That's massive.
Yes. Well, and we've looked at them beyond that too. So they can go on to 100 days too, but for the data we have here, we only showed 60 days. But yeah, we were pretty excited about it. And we think that this is due to the activities of our oncolytic virus. There's something specific, something special about how oncolytic viruses boost T-cell responses that allows for this dramatic effect. And so we demonstrated that by testing out other vaccines. So we wanted to know, is this effect specific to our oncolytic virus? Or is this just a effect that you would see because the vaccination of mice after T-cell transfer is just going to give you such a robust T-cell response? So we first actually tested to see if a non-vaccine oncolytic virus could have the same effect. So this is a VSV just empty, so there's nothing inside of it. It's empty, and we saw that there is very little benefit for this therapy, and it definitely does not show the same as the vaccine effect does. And then furthermore, when we tried to combine our ACT with a peptide vaccine, which is basically just that same protein injected with some inflammatory markers into the mice, and as well we used a so this is an adenovirus vaccine, which some of the COVID vaccines are in the same backbone. So it is also a virus, a vector, but it's not oncolytic. This one in particular is not designed to replicate in the tumor. So we can show here that with a vaccine, you can have some effect, but it's very inconsistent. So again, it doesn't have that same 100% survival that we see with the oncolytic virus vaccine. You have some benefit, but it's very, very inconsistent, very restricted to one of these 5 mice.
Do you think it's the endogenous danger signals that are coming out of when you kill these cells with the oncolytic virus?
And the localization too. So very good segue. So we think that the reason why we have such a consistent effect is because we have such consistent recruitment of those T-cell responses to the tumor. The difference in magnitude of T-cell response, the number of T-cells that we're able to induce by vaccination, wasn't all that different, but the amount of T-cells we see in the tumor, and specifically in the core of the tumor, is very different between these different vaccine systems. So when you use T-cell transfer alone, you can have T-cells that arrive. So this is a whole tumor with the outer edge of the tumor here. So you see some T-cells on the periphery, but if you look at the center of the tumor here, the core, there is very few T-cells. And that is consistent in both the ERK peptide and in the adenovirus ERK systems. But when you look into the VSV ERK, the oncolytic virus vaccine, you see more T cells on the periphery, and there's actually clustering of them around specific sites. So that almost looks like they're just having a fun tumor killing party there. But they also infiltrate into the very core of the tumor here quite well. So we think that this is part of the reason why a large component of the consistency we see with the vaccine, or the tumor regression seen with these vaccinations. You need to have that intratumoral infection and release of inflammatory cytokines and inflammation within the tumor to recruit those T cells back to the tumor and drive this infiltration. So we just wanted to also comment on the length of tumor-free status. So like I said before, the tumor regression was maintained up to 60 days post-infection or post-treatment. And although the tumor regression was correlated with high response here at day 5, we do see a retraction or a collapse of that T cell response down to a smaller level here, although it's still about 10% of the T cells in these mice, which is a pretty good response. If you were to get an influenza vaccine, you would not see the same level of T cell response in a human, and that still provides you very good benefit in terms of protecting against influenza. Now, these are mice; they're different from humans, so we can't completely compare. But this is a very nice and prolonged T cell response that we see with this combination therapy. And so we wanted to see if this maintained T cell response, the persistence of this T cell response, had a good anti-tumor effect after the fact. So we took these mice that had been treated and tumor-free for 60 days, and at least 60 days, and challenged them with another dose of the CMS5 tumor cells to just see if this persisting T cell response could have a protective effect against a subsequent challenge. So say you could prevent against relapse, which is a very common problem in terms of cancer immunotherapy, or in terms of cancer therapy in general. So when we did that, we saw that naive mice, so these are mice that had not been treated previously, died after about 20 days due to the tumor growth, whereas our survivor mice, the mice that had been treated previously and had regressed the tumor for at least 60 days, were able to, there was no development of a tumor in these mice at all. So those tumor cells that were implanted were completely rejected and destroyed before they had a chance to even take hold and grow into a tumor. So that just shows that the long-term functionality of these T cell responses is quite extreme. But what was interesting is that when we looked into the transfer versus endogenous status of these cells, it's quite clear. So this is here a graph showing the kinetics of the T cell response as we showed previously. But over here, we're showing if you take that T cell response and divide it based on Psi 1.1 versus Psi 1.2, which again is transferred versus endogenous T cells, the initial high T cell response is driven by transferred cells, which is not surprising. But the prolonged T cell response after the fact is driven by endogenous T cells. So our transfer T cells seem to have just disappeared, and the torch has been passed on to the endogenous T cells, and they are the ones that mediate this long-term resistance to tumor growth, which is interesting in that the rest of the adoptive cell therapy field has defined that in the context of effector T cell transfer, the persistence of the transfer T cells is very well correlated with the activity and the lack of relapse or the functionality of those T cells. So if you have the more your T cells persist, the better a patient does with the therapy in general. So we're seeing is the opposite here with the die-off of our transfer T cells. We see a better anti-tumor effect, but that's only because our endogenous T cells have come to take their place.
Do you know if those adopted cells are just dead or if they are inactivated or exhausted? What's happening to those after that time?
Very good question. So we have, again, this is one of the figures that I took away to make it more more accessible. But so we did check that, and we looked in and we found that these T cells are going to the tumor and then over time dying at that tumor site. So we don't know yet if they were dying due to the tumor or if they just had done their job and were dying just because they were exhausted, tired, and completed their job. But we do know that when they got to the tumor, that's where they were dying. We checked other tissues to see if maybe they had gone to other tissues and hidden out there, but there was no evidence of them in other tissues. So it really seems like these transfer T cells went to the tumor and kind of, killed the tumor off and then took the rest of the day off.
Did you guys check the bone marrow for these.
Yes,
Okay.
it is. Yeah. Actually, I have this. I have that figure later on. I can show it to you after the fact if you'd like to see. But the point being is we want to make sure that it was actually these endogenous T cells that were having this anti-tumor effect. To see if in the absence of endogenous T cells we could see anything different, if the pattern was different. So for this we used NRG mice, which are genetically engineered so that they're deficient in all endogenous T cells. They have other deficiencies too, but they have no ability to create any kind of T cell response.
They're
So
not
they're
rag
still able.
knockouts, right?
They are. So
Oh, they are
the
rag
R stands
knockouts.
for rag knock. Yeah.
Okay.
So
Okay.
they're nod background, rag knockout, and gamma chain deficient too.
Okay.
So they're completely absent. They're missing B cells too, so that's a slight complicating factor. But since we know that our phenomenon is very T cell driven, we used the NRG mice for this study.
Are they missing NK cells, or the NK cells are just non-responsive?
Yes, non-responsive. So yes, so it would be
Ah.
actually the reason why we use NRG mice here was kind of because we at the same time we were doing another study that required NRG mice, and this question came up, so it'd be interesting to see what would happen, and the NRG mice were available, so it was more of a convenience thing. But we still know that these T cell responses driven from our transfer T cells are completely valid in NRG mice. They're very functional. We get the same level of T cell response, and so we were comfortable using them for this analysis.
You said they also like gamma chain, the common gamma
Yes.
chain. Okay, so
Yeah,
just for background for everybody, CD132 or the common gamma chain, that's an adapter for a lot of cytokines, but mostly include for IL-2, IL-15, IL-21, and some of these are required for T cells, and IL-15 is also especially required for NK cells. So is that possibly
Yes.
why they have unresponsive NK cells?
Yes, so we did do some NK cell depletion, and saw there was some effect. So it's hard to separate the immune response into just one cell components, because they do have a feedback effect. So depleting one population can have an effect on the other population through a side effect, a bystander effect. So that we saw the most consistent effect when we depleted only CD8 T cells. So where we took antibodies that killed off all CD8 T cells and only CD8 T cells. But yes, it's right. It's very hard to separate all the different components of the immune response, because there is a lot of crosstalk between the different components. So it's possible that NK cells could be part of the initial death of tumor cells, which then allows for those tumor-specific T cells to be in the lymph node for our oncolysis to mediate stimulation of them. But using NRG mice, we're able to show that when you treat them with our combination therapy, although you see the same initial regression that we saw in the normal mice, this is quickly followed by relapse, which is interesting because it implies that the endogenous T cells are not responsible, like we suspected for the initial regression, but are responsible for preventing relapse of these tumor cells. At the same time, we saw that the transfer T cells were better maintained in these mice because there was no competition, probably, with the endogenous T cells, which implies that this regression here is not driven by a lack of T cell response, but it still occurs in the presence of ERK-specific T cells. So we suspected that this relapse here was not due to anything associated with a lack of T cell response, but was due to these tumor cells finding a way to get around that T cell response, to find a way to subvert that T cell response. So we first isolated those relapse cells. So this is a little model to show that. We have a CMS5 tumor that's initially regressed by T cells, but there's some cell population that is able to bypass or get around or somehow avoid being killed by the T cells. Although it's a very small population, as the other T cells or other tumor cells are killed off, these tumor cells then are allowed to emerge and regrow out as a relapsed tumor. So we generated a cell line from these relapsed tumors by just taking the tumor out of the mice, putting them into culture in a dish, and growing them up to then use for future studies. And we called them the CMS5 relapse or CMS5R cells. So we first noted is that, as we expected, these CMS5R cells are resistant to ERK-M vaccination. So when you treat them with that same DUC18 T-cell transfer plus VSV ERK-M treatment, CMS5 tumor-bearing mice show very robust regression, whereas the CMS5 relapse cell tumor-bearing mice grow out very quickly. So they seem to be completely resistant to any kind of ERK-M vaccine treatment, which implies to us that they may have an issue expressing the ERK-M gene. And so indeed that was the case. We looked at the genome of these cells. And so as we all know, every gene that you have two copies, one allele and another allele. And within the CMS5 cells, you have one functional allele, which is this A here, and then one mutant allele, which is the C here. And in the CMS5 relapse cells, we've lost the C at this location, and we only have an A at this location. So we can tell genetically that the CMS5 relapse cells have completely lost the ERK-M gene, the ERK-M target. So that's the reason why they're completely resistant to ERK-M specific vaccination therapy. But we have to assume that the same thing is going on in wild type mice, and the same thing is going on in our regular mice, our normal mice. So the question then becomes, why are normal mice able to suppress the outgrowth of these relapse cells? Why are they able to suppress the outgrowth of cells that are not expressing the ERK-M gene? So to further confirm that the only issue with these CMS5 relapse cells is that they've lost the gene, we re-engineered them to express the gene again through a lentiviral transduction. So by doing this, we could then make sure that they express that ERK-M mutation at a very high level. And if you co-culture the CMS5 or CMS5 relapse cells or the CMS5 relapse LV-ERK, which is the engineered ones, the ones that engineered to overexpress ERK-M again, with DUC18 T cells at different ratios, so increasing amounts of T cells here, you can see that the CMS5 relapse cells are not susceptible to killing. So there is no death of CMS5 cells or CMS5 relapse cells in this co-culture system, but the parental cells are killed quite well, and the CMS5 relapse LV-ERK cells are killed off even better. So that's probably likely just due to the fact that we're expressing the ERK gene at a little bit higher level, but it does confirm that there is no issues with these T cells downstream of the ERK or upstream of the ERK. It's really just the lack of that ERK-M gene that is causing these cells to relapse in the NRG mice.
And is MHC expression, is MHC expression maintained in all the cell lines? MHC1?
Yes, so the only reason why we'd have the ability for them to be killed here is if they still had MHC expression, because the only way that that ERK-M would be presented and therefore used as a target by T cells is through MHC presentation. So that's part of why we did this study, to confirm that, yeah, the presentation was not an issue here.
Yeah, so they can still present other tumor antigens regardless of having ERK-M or not.
Yes. So thank you for that segue. So knowing that endogenous T cells are very important for preventing this relapse, but knowing that ERK-M responses, whether it's transfer or endogenous, have no effect on CMS5 relapse cells, we thought that maybe the mechanism that we were seeing was due to development of T cell responses from that endogenous compartment against other tumor-specific targets. So luckily, a recent study by Dr. Pramod here profiled other antigen mutations within the CMS5 model and showed these 4 here are specific mutations expressed by CMS5 cells that are tumor rejection antigens. So that just means that if you develop a T cell response against them, you have a very high likelihood that the tumor will then be regressed by those T cell responses. So we took a pool of these peptides and stimulated blood cells from our treated mice to see if they have developed T cell responses against these other neoantigens in a mechanism called antigen spreading, but I'll discuss that in a second. And so we saw that when you compare to BBS controlled and ERK peptide vaccinated versus ERK peptide vaccinated mice, you saw some increase in those responses, but it was not statistically significant different. Whereas in the VSV ERK-M treated mice, we did see a significant shift in the level of T-cell responses against those epitopes. So that just tells us that spreading, so that means that during the process of our treatment, although we've developed a very robust ERK-M response, we've also developed other bystander responses that have a very good chance of killing off tumor cells, irrespective of whether they express ERK-M. That's very pronounced in oncolytic virus vaccination, and more pronounced in oncolytic virus vaccination than compared to other vaccine platforms. So this, in addition to driving infiltration of T cells into the tumor, comes back to your question earlier about why oncolytic viruses work so well. I think it's also because they're able to augment the antigen spreading effect. So that's our hypothesis, that oncolysis is what mediates this, but we haven't completely confirmed that yet. So that leads us to our model here, where we know that with oncolytic virus vaccination, we can simultaneously drive death of tumor cells and exposure of those antigens to DCs, or through DCs to T cells in the lymph node, as well as direct tumor vaccination or anti-tumor cell vaccination in the lymph node through our direct infection of DCs by the oncolytic virus vaccine. So that results in a two-round attack where you have the transfer T cells here driving the initial tumor regression, and although you have outgrowth of antigen escape variants, so that being those cells that have lost that ERK-M gene, the spreading effect driven by oncolysis can then drive development of T-cell responses beyond that ERK-M specificity and allows the development of responses that can eliminate these escape variants, irrespective of ERK-M expression. So, like I said, I think we like to contribute the special ability of VSV to drive regression and lack of relapse to its ability to recruit T cells and infiltrate those T cells into the center of the tumor, and that is by doing this with transfer T cells, they can drive initial tumor regression, but this regression causes selection for an antigen loss variant. If you kill off all the cells that are expressing ERK, then the only cells that can survive are the ones that have lost ERK expression. And it's only through this in situ vaccination effect, which is again that oncolysis-driven vaccination effect, can you stimulate an antigen spreader response to eliminate all those lost variant cells. So the real question at the end of this is, what is the clinical significance of it? This is all in a mouse model. Is there any way that the lessons from this study can be applied to human therapy in the clinic? So that brings us to this preconditioning idea. In a lot of ACT therapies, because they know that the transfer T cells and their persistence is important, and that persistence is truncated by the presence of endogenous T cells through a competition mechanism. You know, if there's only a certain amount of nutrients in the environment, then if you have less cells to compete with, then those transferred cells can compete better and survive longer. It's common that in ACT therapies, that we use a preconditioning regime which depletes endogenous T cells, removes them from the system, and therefore prevents them from competing with our transfer T cells. But in our model, it would stand to reason that this preconditioning might actually prevent the antigen spreading phenomenon from happening, and therefore prevent the anti-relapse effect of our therapy. So to test this, we followed up with using normal mice again to kind of recapitulate what we saw with NRG, using a CD8 depletion regime, or by depleting only endogenous T cells using our Thy1.2 antibody here. We're able to see that, yes, indeed, you do see very rigorous relapse of those tumor cells. And then furthermore, over here, the CPX treatment, it's a very common drug, cyclophosphamide, that has been used in the past, although not so much anymore, as a preconditioning regime. So by using this CPX drug you can deplete the endogenous T cells to then allow those transfer T cells to survive longer. So when you do that, you do see even better tumor regression because CPX is also a chemotherapy. So this tumor is attacked from several different angles. But although this regression is robust, it then does ultimately result in relapse of the tumor. So we think that by preconditioning these mice, we've actually predisposed them to experiencing a relapse due to the lack of antigen spreading. And that's, again, prevented by the lack of endogenous T cells in these mice. So we think that should be taken into account moving forward for developing clinical therapies for cancer. And this is a little model of how that happens. So again, you have the initial tumor regression driven by the transfer T cells and oncolytic virus vaccination. But if there are no endogenous T cells for the subsequent lysis to provide antigen and secondary stimulation to for responses beyond ERK-M, we end up seeing a very robust growth of those antigen negative variants and relapse. So that brings us to the end of that story, but we do still have some unanswered questions. So as I alluded to before, we think that the antigen spreading mediated by LVV is oncolysis dependent, but we haven't completely confirmed that. So I'd like to do that at some point. And the responses we saw were quite low. They're only 1 or 2%. So looking into ways that we can change that so that the oncolytic virus vaccine can cause the tumor death in such a way that it is more inflammatory and therefore more likely to induce spreading to other antigens. So I'm pursuing this via collaboration with Dr. Sam Rookenhee, who was a research associate at McMaster in the Mossman lab, but has since moved on to work as a PI at the University of Guelph. Secondly, it's interesting that we see such pronounced antigen loss. It's very consistent, the loss that we see. It's in most treated mice that don't have endogenous T cells, we see this relapse happen quite consistently. So we want to figure out what is the mechanism for that antigen loss variant and how do they come about? Is this a pre-existing heterogeneity, or is there something about our therapy that helps to induce this loss? And so my favorite hypothesis for the moment, although it still remains to be tested, is that inflammation tends to induce a lot of reactive oxygen species, which is not nice to the genome. These reactive oxygen species like to go in and cause a lot of damage to the DNA, and the result of that could be what results in this antigen loss variant. As soon as you cause any kind of DNA damage, the genome becomes so unstable that you could be losing bits of genes everywhere, which could include our ERK-M gene. So is it possible then, if it is DNA damage that's resulting in this antigen loss, can we use drugs to prevent that loss and thereby remove the onus from the oncolytic virus vaccine to spread the response to other epitopes? So I'd like to acknowledge the people who worked on this. So Dr. Young-Hong Wan, my advisor, for being such a good advisor as well as tenacious advisor. And Boris Simovic and Dr. Lan Chen and Donald Baston were all contributors to this research, and the rest of the Wan lab as well. And our collaborators, Dr. Jonathan Bramson, Dr. Brian Lichty, and Dr. Kyle Stevenson, as well as Telvir Mendur for helping us do this research. And Dr. Lise Norian from the University of Iowa provided us with the Duck 18 mice, so we are very appreciative of that. And finally, the funding. All these various sources were contributing to this research. So thank you. I'll take any questions you got.
That was a very nice presentation. It's got me so excited. But wait, before I go on shooting questions, do you guys have anything?
Thank you for the nice presentation. I have one question because you were mostly concentrating on solid tumors. And I was wondering if we could maybe in the future use it also in cancers like myeloma, lymphoma, or leukemias. What do you think about it?
Yes. So the reason why we focus on fibrosarcoma is because we know that other cancer immunotherapies have had a lot of success treating melanoma and liquid tumors, but the solid tumors are the ones that seem to be most resistant to cancer immunotherapy. So we want to directly address the harder instance of cancer therapy, given that melanoma has already had a lot of success. And that's also probably associated with their lack of solid structure. So if you have a liquid tumor, you don't have as much issue getting infiltration of the tumor because the tumor is fairly wide spread out and will actually traverse as it goes around. Whereas this fibrosarcoma has that really dense structure, and it's more difficult to get the tumor infiltrated with T cells because of that structure.
Right, thank you.
But yes, my previous JCI paper was looking at a melanoma model, so we know that it does work. This combination therapy does work in melanoma.
And have you looked at metastasis? Does it work in at metastatic sites as well?
We haven't seen any evidence of it, especially because we have taken these mice out to 100 days, and you'd think that if there was any kind of metastasis that wasn't being controlled by the ongoing T-cell response, you'd see that outgrowth eventually, and we see no evidence of it whatsoever.
I know that you did do the same therapy in the B16 model, which is a highly metastatic one, so you didn't see metastasis in that either.
So no, not as yet. It seems like the B16 model is a little bit more genomically unstable, so we do see relapse more common with them. So that is an issue there. But the mice that we were able to completely cure without relapse at the primary tumor site, we saw no evidence of metastasis in those mice, and we carried them out for the same extended time, about 100 days.
I have a second question, which is more, I would say, just like a very broad general question. How difficult do you think this would be to, actually employ in the clinic? Like, do you think that there would be some sort of like off-the-shelf product? I mean, I know for ACT, you generally want to take TILs from the patient because they're cognate to their MHC and their antigenic landscape. But do you think this would work, for example, with CAR T cells?
Yes. So there's actually a student in Jonathan Bramson's lab that is studying that right now to see if CAR T cells can experience the same kind of boost from an oncolytic virus vaccine or a VSV vaccine. And I don't want to take the liberty to share any of her data. I'm not too familiar with it, but I think she's seen some success. So it's entirely possible to extend it there, although things will be different, right? Because you're not talking about presenting anything in an MHC anymore. You're talking about expressing it as a cell surface protein. So there are differences there, but I don't think they are differences that will be that will prevent the efficacy of the therapy.
Samriti, is that it? Are you?
Yep, that's it. Thank you.
Okay.
Thanks for answering that.
Okay. There was a study in 2020 that wasn't, I wouldn't say similar, but it ended up with a very similar conclusion. What they did was they had adopted cell transfers, but these ACTs, while being against, so they used the HER2 model, HER2 breast cancer model, and while these TILs, adopted cells, were against HER2, these cells were also carrying a membrane-bound growth factor for dendritic cells. So what they found was so I think they used CAR Ts, and while these CAR T cells were killing the target tumor, they were also enhancing the host's own dendritic cells, which then in turn would augment the T cells within the host. And they found that when they added just HER2 specific CAR T cells, there were host T cells that were against other antigens on the tumor, which were able to suppress the tumor much better. So this is similar to what you've shown here about epitope spreading. And it looks like without the help of the endogenous T cells, there's just as much as you can do with your external therapies.
Yeah. Well, and so I think what they're pursuing again is similar to what we're pursuing, in that although they're using engineering of those T cells to mature those DCs, we're using the virus to do that.
Yeah.
So that same inflammation that's induced by the initial round of virus infection will provide those same growth factors, the same maturation factors to the DCs, and then ultimately drive development of those T cell responses. And the T cell attack in their case will also result in the same lysis and exposure of tumor antigens, tumor neoantigens to those DCs to allow for the presentation in the lymph nodes.
Yeah. It's funny how things like epitope spreading that are seen from a negative perspective in autoimmunity, because they're very bad for autoimmunity, and you can't even you start with one autoantigen and now you have several of them. But in cases of viruses and cancers, they're just so good. One more thing I want to ask you about your hypothesis about the ROS. Do you think the ROS coming from the immune cells are going inside the tumors and making it unstable?
Yeah. So I don't know for sure.
Mm-hmm.
There was a study a couple years ago that looked at DNA or antigen loss variants due to ACT. So this was in a OVA specific model, but the conclusions was that interferon gamma produced by the T cells was causing the induction of ROS and therefore causing the DNA damage, resulting in loss.
Oh.
In our case, it could be stuff coming from the T cells, inflammation, cytokines, all resulting in ROS production by the T cells. But at the same time, our oncolytic virus also, or other oncolytic viruses, as well as ones of the same family as VSV, have been noted to induce ROS at the tumor. So initially, that seems like a good thing because ROS can cause DNA damage, and DNA damage can result in death of tumor cells. But if that ROS is not enough to cause the death of all those tumor cells, then that DNA damage might come back to bite us by causing this kind of antigen loss variant arisal. So yes, it's all interconnected. It's just trying to figure out what is the best way to approach it. Can we make it more without making it too much to the point that it becomes a bad thing?
Yeah, I have another thing to add. What if it's because these tumors, they grow in such high density that they have to rely on high glycolysis instead of they can't
Yep.
possibly have so much oxygen, and in the end they end up with very messed up mitochondria. And when mitochondria they don't work, they also release a lot of ROS. So there could be an intrinsic source of ROS within the tumors.
Yeah, so that's what I was saying before. Then maybe it could be a pre-existing heterogeneity. It could be that the source of the loss can be at several different steps. So it may be better that trying to stop the DNA loss or the DNA damage and therefore antigen variant loss is not a viable strategy. And so what we need to do then is focus more on inducing more of those antigen spreading responses. So I think by tackling it, by using both those criteria, we can get a better understanding of the entire phenomenon and therefore better address our treatment strategies to best suit our results. So yes, trying to strike a balance between the two is probably the best thing to do. To figure out if one is unaddressable, then we move on to the other one.
Yeah. Just a general question. Does this happen to anybody here that you read a paper and you get so hyped that you really want to talk to the author? You wish you could have a chat with the author for 5 minutes just because that thing is so interesting?
Yeah.
That's what I had with this paper. So I read your article after we invited you because Samriti had some interesting stuff. I had read the abstract and I was like, this is pretty interesting. And when I read the whole article, I was so hyped with this paper. I'm really glad we got to interact with you, specifically me. I'll be very selfish here. I'm very glad I got to interact with you and got to know about this very cool stuff.
It's a great opportunity for me too to present the data too, because you don't get a whole lot of feedback by publishing other than from reviewers. So it's nice to get more feedback from a wider audience.
Yeah,
and it's nice to know people reading it too.
Yeah, there should be some comment section under publications. You have a moderated comment section where people can say what they just saying, hey, I loved your work. It boosts the author's morale so much.
Well, places like ResearchGate provide that kind of function.
Yeah.
Don't take advantage of it enough.
It's very easy to implement. I'm sure if journals want to, they can all do it for their own articles. And there are just few people who are on ResearchGate. There's still a lot of people who don't know about it. So
Exactly. Yeah.
yeah, something like that. A discussion forum would be great for journals. Maybe that's what the new generation of journals will be. They'll allow discussion on a paper right below it. Like even in a paragraph, you
There's a
could
lot of preprint servers now too. I think, like I said, I think the preprint servers have some of those options available. I think there is some discussion, some message boards allowed on those.
it's going to be the future. Yeah. So do you guys have anything else to add? Okay, Dr. Scott, thanks a lot.
Thank you. Yeah, thank you for inviting me and allowing me to share my research.
Yeah, just last thing, not research related. You told us that you like listening to audiobooks.
Yes. Yeah.
Which audiobooks do you listen to? And is Antibuddies one of those?
That's not audio.
Well, the
Product placement.
audio, I haven't ventured into podcasts yet, but I'm running out of books, so I think it's about time that I start, listening to some podcasts in addition to my audiobooks. It's really easy to do, especially if you have an hour drive every day. You can get a whole podcast done in one go.
Oh yeah, that was one of my inspirations initially to start with. I was driving to places and I was running out of NPR podcasts to listen to. So, anyway, thanks a lot, Dr. Walsh, for joining us today. This was a very good discussion.
Thank you.
Hope you have a nice 2021.
You too. Bye, everyone.
Thank you. Bye-bye.
Thank you. Bye.